All right, thank you guys all so much for coming out to this semester's Brain Science Speaker Series. This is organized by LSU Mind and hosted by CCT, which we really appreciate. And so the mission of LSU Mind is to coordinate and strengthen research, training, and community outreach opportunities here at LSU. My name is Heather Lucas, and I'm on the executive committee of LSU Mind, along with Melissa Beck, Mark Galecki, and Susan Duncan. And it's my great pleasure to introduce today's speaker, Dr. Robert Hampson. Dr. Hampson's visiting us from Wake University Forrest School of Medicine, where he's a professor of physiology and pharmacology. His work focuses on understanding how memories are formed in the mammalian brain, as well as developing new ways to detect, manipulate, and even restore memory abilities in people suffering from memory loss. His work has been funded by multiple centers within the NIH and most recently through DARPA, or the Defense Advances Research Project Agency, and his work has been cited over 11,000 times. And if that weren't enough, he's also a prolific science fiction writer, and his sixth novel? Fifth novel called The Desert and the Sun is coming out sometime in the next year. Is that right?
March.
In March. Oh, okay, in March, right? So you can mark your calendars to check that out. Right? And so as an interdisciplinary initiative, LSU Mind is very excited to be hosting Dr. Hampson and to have the opportunity to hear about his exciting and highly interdisciplinary work in the area of neuroprosthetics. Okay, so please join me in giving Dr. Hampson a warm welcome.
Thank you, Heather. I want to thank LSU Mind for the invitation and to thank Heather and Mark and Melissa and Chris who've hosted me so far. I've had some wonderful meetings today. Started off a meeting with the graduate students, which was a lot of fun, and I really enjoyed doing that. I'm an old professor, okay? We'll start off with that. And as I look at a career in research, I find myself gravitating to teaching. And so I have a strong commitment to reaching out and shaping, training, and encouraging the next generation. So I'm very glad that we can do that. And I understand that we are streaming the talk, which is also wonderful, and recording it, because I'm going to take it back and I'm going to show it to my own department and grad students and say, why aren't we doing more of this? But again, thank you so very much for the invitation. Now I'm told I have a commanding presence and a lecture hall voice. So I'm going to pace. There will be times when I pace and times when I walk back and forth. Now, the stand and lean, I like to look out at the audience. I like to move back and forth so that I can see all the faces, and try to hopefully make some eye contact with folks as we go along. So if I'm ever standing in front of what you want to see, you can raise your hand and say, Dr. Hampson, please move, or you say, Dr. Rob, or Rob, would you please move? That will be fine. The other thing is, don't feel that questions have to wait to the very end. If there's something you don't understand, and I can hopefully explain it quickly to get it so that you can continue following, go ahead and ask the question. We are going to have Q&A later, but go ahead and ask the question, because if it means you stay engaged, I'd much rather work with that to start with. So the question is, human memory, can it be fixed? I started off in a laboratory studying place cells in the rodent hippocampus. And place cells are very interesting. Place fields are very interesting. It's a part of the brain that interacts with our environment without being a sensory structure. And yet there's something cognitive going on, and there's something memory going on. And So many of my colleagues and the experts in the field at the time I started this study in the early 1980s were saying the hippocampus does place and that is it. And yet we know that's not the case. I've been told I need to not forget my fiddly bits, which are my funding sources and my various partners. Most importantly, I always bring up neurology and neurosurgery. They very graciously granted me a secondary appointment in neurology so that I can work closely with the clinics. But also colleagues at University of Southern California who are deeply involved in a lot of this work, Rancho Los Amigos, Virginia Tech. And as a COI disclosure, I am a consultant and co-founder with a company that would like to take some of these principles into a medical device. So that's my disclosures right off the bat. Key problems in human aging. Remember what I just said. There's something going on in the hippocampus, and it has to do with memory and aging. So what are our problems as humans? We're susceptible to disease. We have reduced mobility. Oh my goodness, I know that. Bad hit. Dementia, memory loss. Where am I? What am I doing? We have a number of enabling technologies. We have pharmacology. We've got great drug development working at various disorders of aging. We have the possibility of gene engineering. At the very least, we can gene engineer the tools that we use for our drug discovery or for our drug production. But we have a growing field of neurotech. Can we exercise precision medicine to the point of delivering a treatment, delivering some sort of a stimulus, an inhibition, an excitation, something directly to the part of the body and the part of the brain in which there is a disorder. And so I'm going to talk about that. Memory loss, dementia. It's predicted that it's going to hit 50% of all adults by the age of 90, that reach the age of 90. But 11% of adults that are aged 45 and older self-report issues with their memory. And we have other diseases that affect memory. It's not just Alzheimer's disease, but epilepsy, Parkinson's disease, Huntington's disease, Korsakoff's disease, traumatic brain injury, stroke. All of these are diseases and disorders that impact our ability to remember. And in particular, they impact our ability to form and create and store and retrieve new memories. Now, most of what we understand about human memory started with the case of HM. Henry Molaison was a young man who had intractable epilepsy. His epilepsy would not respond to the drugs of the time. It could not control his seizures. So William Scoville, his neurosurgeon, resected the medial temporal lobe, both sides. And he largely affected the hippocampus, but we also know now that it also took out large chunks of perirontal cortex, entorhinal cortex, the amygdala, but in particular the hippocampus. These are the areas right here. Here's hippocampus. These are the areas that HM lost. And from that day on, HM was unable to create, store new memory. If you showed him a newspaper, he would remember what he'd read for about 10 minutes, and only about 10 minutes. After 10 minutes, we lost. He'd never seen the newspaper before. He'd never been able to, he didn't know what the items were. He didn't know any of the information that was new to him after the surgery. Before surgery, he did have a number of those memories, but they were affected by the fact that he had a seizure disorder, but he did have memory. From this, we know that hippocampus is involved in anterograde amnesia, amnesia from the point forward. And he was having difficulty. Very strange and unusual phenomenon was that procedural memory did not appear to be affected. He could work puzzles. He got better all the time at puzzles, although he would tell you he'd never seen one before, a particular puzzle. But he would be able to work the puzzle as if he's familiar. Remember what I said. I started off studying the hippocampus in rodents. And at the time, we were told the rodent hippocampus does space, nothing more. But we know that that's not the case. We knew that from HM. So this has led us into a number of different studies, and I spent way too many years studying rodents, and I spent quite a few more years studying non-human primates, and now for the last 10 years have been studying and working with humans. So the tally is 40 years less, let's see, shut down, okay. So call it 35 years with rodents, 20 years with primates, and about 10 years with humans. That adds up to way more time than I've actually been practicing. There's a little bit of overlap in the middle. But one of the things that we're looking at, and what's fairly key to where we're going now, is the human memory circuit. Well, James Pepez proposed a circuit for emotion that we now know is, yes, it's involved in emotion, but it's not the sole purpose. And it's very heavily involved in memory. And it involves, starts from the nucleus basalis and minor. It also includes the fornix, comes into hippocampus, also includes the mammillary bodies, the mammillothalamic tract, the entorhinal cortex, and the amygdala. So it's a fairly complex network, and it forms a loop. Now you'll hear a number of people refer to this as the limbic system. Well, it's a little bit less than the limbic system. We talk about the Pepez circuit. It's a little bit more than the Pepez circuit. But the human memory circuit is fairly complex, and again, it also spreads its influence out and involves the prefrontal cortex, the cingulate cortex, and of course we have connections back to temporal, parietal, occipital, frontal. It's everywhere, and it has an awful lot of implications. Now, one of the questions that came up because of the medical treatment of deep brain stimulation for Parkinson's disease is, hey, we can stick electrodes in the human brain and we can tickle this spot and look and see what happens. And if you tickle the hippocampus, what happens? Sometimes you trigger memories, and people will report a memory. Just a patient anecdote. After a test one day, came back into the patient room to talk with the patient the next day. She said, hey, after that session we had, I had this memory of riding my bicycle at my grandparents' place. I haven't thought about my grandparents in years, and I remember this whole thing. And did we do that? No idea. But it's the sort of anecdotal evidence that we got, and there has been non-anecdotal, actual documented evidence of stimulation in parts of the brain, such as fornix, such as the hippocampus, triggering recall of memories. But that's recall. That's not storing the new memories like we just talked about hippocampus in the case of HM. So the question is, will stimulation work to help boost memory, to help restore a function when that function is being lost? And if stimulation works, where in that memory circuit do we want to stimulate? Well, in answer to the first part of the question, Laxton and Lozano in Toronto applied a DBS-style stimulation to the fornix. And what they found was, hey, the We slowed the rate of cognitive decline, slowed the decline in several biomarkers, improved glucose utilization and blood flow in neocortex, and there were no serious adverse events. This has now been spun into what's called the ADVANCE-2 trial. Laxton and his colleagues at Wake Forest have recruited, consented, and operated on approximately 1/2 of all of the patients of the ADVANCE-2 trial. And the biggest result that they're getting so far is the metabolic improvement and a slowing of the decline in the mini mental state exam, the MMSE, which is considered the gold standard for Alzheimer's dementia. And with the MMSC, there are a number of things having to do with being able to recall word lists, being able to draw pictures from memory, and one of the most heartbreaking bits when you're working with patients being assessed this way, patients asked to draw a clock face, a round analog clock face. Put the numbers on it, put hands, maybe yes, maybe no. Person who has an MMSC of over 30 can draw you a nice clock face, symmetrically placed numbers in the correct order around the face of the clock. They may or may not put clock hands on. When the MMSC drops down to about 25, they start leaving numbers off. Down to about 20, they cram all the numbers onto one side of the clock face, and they may and probably leave about half of them out. When they go down below a 20, they're lucky if they can draw a circle. So there are a number of things that we look at. So being able to slow the decline of the MMSE is a great thing. But this is what other people were doing. Question is, what can we do as a precision measure to slow the decline, to improve the memory or restore memory function? Well, I was working as part of the DARPA RAM program, Restoring Active Memory. And what DARPA's goal was, can we demonstrate a stimulation effect that will improve memory, facilitate memory in cases where memory is lost? 2 approaches finished that program. One led by Dan Rizzuto and Mike Conant at the University of Pennsylvania, and they put together a fairly large multi-institutional group which they actually looked across large portions of brain and said, what is the brain state across large real estate that is associated with correct memory? And is there a place we can stimulate to restore memory? And they found one, and they did it, and they showed an improvement in memory. The other approach that came out of this program is the one that I was involved with, and we were prime contractor to DARPA with our colleagues at the University of Southern California, in which we created a MIMO model, a nonlinear dynamic model of hippocampus in and out. Neural patterns coming into hippocampus as evidenced by ensemble activity in CA3, and what's the output as evidenced by ensemble patterns in CA1. Now that's a monosynaptic connection, fairly easy to model. Our first stage of the model, just simply modeling CA3 and CA1 with the intent of moving those 2 points out, the input and the output, to a point where we could potentially bypass a damaged hippocampus. And we had a number of great successes with different types of models that we were using for that purpose. Now, the nonlinear model is one that says when a neuron fires an action potential, a spike, if it fires more than one spike, we can calculate the nonlinear dynamics. What is the probability that each time the neuron fired, that this previous firing was in a particular time window? So we look at the interspike timing, and that varies a lot. Think of a dripping faucet. It's not a regular rhythm. Well, the brain is a lot like that as well. And so a zero order nonlinear function would be simply what is the firing rate of the neuron. A first order would be a function of all of these intervals in between each firing. Second order function would look at not just the most recent spike firing, but also, in addition, the one prior to that as well. So we're now looking back 2 events in the past. That's useful if you're wanting to look at only a single neuron, but what if you're wanting to look at multiple neurons? Well, then you need to start looking at cross kernels. So in this case, what is the timing relationship between this spike and that spike on a different channel, this spike and this spike on a different channel? So cross kernels are important as well. So the nonlinear MIMO model was actually built from that. And our brain function is envisioned now as sensory input, producing spike trains in the input to hippocampus, transformed into spike trains in the output of hippocampus, and that translates into something else, whether it's motor control, behavioral control, memory control, it's something else. And our what we were able to do with our models, we model the upstream and the downstream. We like to plot them as color density maps, heat maps for firing rate, in which this horizontal axis is time, the vertical axis is channels, different neurons. And to give an example of how we actually implement this, I'll show you a diagram from primate hippocampus. In the macaque, we would record, sorry, I keep hitting the wrong button. We would record from CA3, get the strip charts, real-time activity, run that through the coefficients of our MIMO model, our nonlinear model. That would give us an output prediction, and then we use that prediction to program an ensemble stimulation, a multi-site, multi-spatial, multi-temporal stimulation pattern. I have no idea if the video is going to run, because this was actually generated for a PC, and offhand I'm going to say that video is not going to run. So, and it is hanging up. Let's, no, come on. There we go. That'll work. Good enough. Fair enough. What the task looks like is 150 to 175 trials in what we call a delayed match to sample task, in which one picture is shown to our patients. They touch the screen. We use a touch screen. They touch the screen to indicate that they're paying attention, that they have recognized that image is up there. And then we blank the screen. After a short delay, we will show them anywhere from 2 to 4 images. We could show them up to 8. We typically show 4. And the patient touches the same image that they saw. We'll run them through our 175 trials. And in those 175 trials, we don't expect there to be any real failures of memory. Very short delays. You know, as humans, we're really good at pictures. We're really good at pictures. Which is a real shame, because in a primate, we can run that delay out to 2 minutes, and the primates don't remember pictures very well. With rodents, we have trouble getting more than just a 2-choice, them to do more than a 2-choice task. They don't like that. Humans, we try to set it up and do it the exact same way we did in the animal model. It isn't going to happen. It isn't going to happen. So what we did is to say, all right, we'll run you through your 175 trials. Take about 30, 35 minutes to do that. Right, take a break. Eat lunch, go to the bathroom, whatever it takes. Our patients are in a dedicated EMU. They can actually get up. There are some places where the epilepsy patients are held in ICU and they're stuck in the bed until those electrodes come out, which is a darn shame because it gets boring. Which is one reason they're very happy to work with us. After that interval, it's now been an hour since they first saw the images. And we'll show them 3 at a time, and we'll say, hey, see these pictures? I'll show you 3 pictures. I want you to tell me if any of them are familiar to you, and how familiar are they? Are you going to tell me, hey, I saw that in that task you ran? That's a 5. I think I saw it, maybe. That's a 3 or a 4. I don't recognize this. Maybe it's kind of familiar, but I'm not sure. That's a 1. I haven't seen this at all. That's a 0. Well, if you take a look at these 3 pictures I put up here, one of them was that target. They saw it here, and they touched it both times. So that's the target right there. This one is a distractor. We've shown it to them. Yes, they have seen it, but it should not be as familiar as a target. This one right here, there we go. This one right here, that's a foil. That's a novel. They haven't seen that one at all. And that is our control. If they tell me that's the one that's most familiar, we have to throw that trial's data out, because that's a non-target or a false alarm. So we go through that. It's now been 30 to 90 minutes since they have seen the images as a sample and a match in the non-match in the delayed match and sample task. And we test to see how well they remember, how well do they recognize the images that are up there. Now, we also have another variation of the task. We use pictures. And some of the pictures that we use could be categorized. And these are 5 categories that we found fairly early on that appeared to have neural activity associated with them that was above background. So that'll come into play a little bit later. But here's a nice sampling of the different types of pictures that we might see. And we are currently working with a data set of over 3,000 images that we can use that I would say at least half of them have been categorized by survey to say, okay, what's in this picture? And this way, we don't contaminate anything with a picture they've already seen. We are able to run 4 different tests, 3 or 4 different tests with the patient without repeating anything. And that gives us the tool that we need to ask the question, can we create a brain state favorable to memory? Can we find a code for memory in the hippocampus? Or how are we able to control the function? So I mentioned that we're working with epilepsy patients. Epilepsy patients, like HM, who have intractable epilepsy. Nowadays, instead of going in and cutting first and asking questions later, we stick electrodes in, and it's a fairly simple procedure. As little as 10 years ago, that would involve cutting a large chunk of skull, exposing the brain surface, putting electrodes on it, putting electrodes in it. Now we drill a 3 to 4 millimeter hole, thread a bolt into it, thread an electrode through that down into the part of the brain that is important to our function that we wish to record from. Tighten the whole thing up, close everything back up, close the skin, and give the person a turbine for a week. And in that turbine, we'll have connections to all of our electrodes. So, question is, what did we do? Actually, I'm showing you here a couple of pictures. Here's a couple of different MRI images of hippocampus, cross-section of the temporal lobe. This is what the the MRI shadow of our electrode looks like, superimposed over hippocampus. The beads are the EEG electrodes. They're fairly large metal cuffs that go around. And what you can't see are the fine single neuron electrodes that are spaced between them. So I show a little bit of the diagram down here. We have 4 microelectrodes in between the macroelectrodes that enable us to position those near the cell layers in hippocampus, and we can record. And if you can see some of these shadows on here on this CT scan, I'm afraid when my laser goes up against the black, it gets lost. But you can see the shadows of the electrodes coming in here. Here's our hippocampal electrodes. There's an amygdala electrode, a couple of prefrontal electrodes, cingulate, and so on. That would be some combination of which most of these patients are going to get. The neurologists are going to record the electrocardiogram, trying to figure out where the seizures are occurring. But once the patient has the electrodes in, it takes a little while to taper off the anti-seizure meds. And before they have the electrodes taken out, they're put back on the anti-seizure meds. During that period of time, those 2 periods of time, we go in and we record from the hippocampal microelectrodes, and we exercise one of our models in terms of how do we stimulate in the hippocampus and generate a change in behavior. The answer is very quick look at what we did. Our very first result, if we record only and look at the behavior, this is a case where we were working with just stretching out the delays in the delayed matched sample. If we did that, we could only stretch them so far, but stimulating certain trials caused them to be performed about 20% better. An improvement in performance of about 20% mean correct. The actual improvement runs to be about 34% of their previous performance rate. In the delayed recognition part, the part that tests the retention 30 to 90 minutes later, we also saw a great improvement. This one was running up to about 37% of the original as an improvement. More importantly, what this told us is that we were decreasing errors. We were seeing between 40 and 50% decrease in the errors that were being committed by these patients under these conditions. Now, I show you the yellow case in which we were just recording only and not stimulating. What did our performance look like? As a comparison, I also show you the non-stimulated trial performance. These non-stimulated trials are interposed between the stimulated trials. So stimulate, improve the retention. Don't stimulate, it's normal, it's control. We also randomized our coefficients in the model and would see that one of 2 effects. Either we see a decrease in performance or we would see no change whatsoever. So here's an instance of seeing a decrease in performance. Here's an instance of no change. So the randomized stimulation wasn't really doing a whole lot. It certainly was not producing the facilitation that we see. Now, I mentioned that we had a couple of different models. The MIMO model is what you just saw the results from. The MIMO model is something of a bypass for hippocampus. Hippocampus is malfunctioning, let's bypass it, or let's support it. But what about the fact that we're giving different information content in these pictures that we show the patient? Is there something about stimulation that is key or interacts with the information content? So we had a second model that started with the MIMO, and we looked at what the model would generate from given images, given pictures, from all the data that we had recorded across 30, 40 patients. And we started looking on any individual patient, was there a common pattern all of the times when the picture that we asked the patient to respond to was an animal, cat or dog, a building, a plant, a tree or flower, bush, a tool. And by the way, this was a misnomer category because all of our survey respondents referred to anything that looked like arts and crafts supplies. They said there's no category for arts and crafts supplies, we'll call them tools. So, in fact, we saw a lot of multicolor and arts and crafts supplies, and then of course vehicles, cars, trains, bicycles, planes. And again, what you see is that overall we see an improvement in the memory retention in the delayed recall part for most of these categories. Now, the overall is kind of low. Why is the overall low? Well, it's this category right here. This is proof that the best possible design will still fail if your assumptions are wrong. Our assumptions were wrong. Our assumptions about what constituted the building category in all likelihood are wrong. So we're kind of stuck with this. We have to explain it away every time. But it's okay because it tells us we're making progress forward, understanding what these possible codes are. But as we go a little bit further with it, we start looking at individual subjects and individual stimulation patterns and categories. And what we find is that it's actually a lot more variable even than what shows up here. Here, the only category we see in which stimulation caused the performance to go down is the building. But when you look at an individual subject, there's a very, very imperfect match between the category of image as we have defined it and the category stimulation. We think that the reason why is because pictures of buildings are not clean pictures of buildings. They often show grass, trees, flowers. They might show people. They might show a car parked next to it. They might have a dog or a cat out in the yard. They're not clean images. So currently, one of the things that is ongoing is doing a deep dive data mining of this data to find out what is it that worked, what are the circumstances in which stimulation worked, and what do those trials correspond to? Could it be blue sky? Could it be green grass? We don't know yet. It's something that we're starting to work with. But it leads us into some interesting questions. When we are stimulating, are we strengthening what's there, or are we writing in something new? Well, how could we test that? How about, instead of showing a person a picture in the first phase of the DMS task, instead we just show them a gray square, but we stimulate them anyway with an image, with a pattern for a particular category, and then we go back and we say, okay, how likely are they to pick? If we stimulate with a pattern that we think should be animal, do they pick the animal picture? And when we test the patient, we don't tell them this is what we're doing. And as a matter of fact, we have a control run that we do to collect some of the background data to figure out what actual probabilities are, which is we say, okay, when you see a gray square, and you will then see 4 pictures, just pick whichever one appeals to you. That does 2 things. One is when we're not stimulating, it gives us a little bit of an insight into natural biases having to do with the types of pictures. When we are stimulating, it doesn't tell the subject that we want them to pick something in particular. But when we do stimulate, we've actually found that the choices are greater than chance. Now you would think we show 4 pictures, that chance level would be 25%. We have 5 categories, so maybe the chance level is 20%. But actually, the way we are generating these images, we don't have just 5 categories. We have a lot of uncategorized images or images that might be possibly categorized as something else. So when we go through and use that data from the controls, where we told the patient to just pick something, we find that chance, random performance, is down here with these orange lines. So if stimulation had no effect whatsoever, that's where each of those bars should be. But they're not. 200, 170% increase over chance probabilities when we're stimulating. So again, are we writing in something new, or are we strengthening what's there? One of the other things that we do now is to take a look at the non-stimulated trial performance. In this delayed recognition, this is the 30 to 90 minutes later, how well is this image remembered? How well did the stimulation work? And we look at the no stimulated trials and these write-in, these ambiguous sample trials in which there was a stimulation, and during delayed matched sample, they picked the image from the category that matched the stimulation, or they picked something else. So we separated those two results into the blue and green bars. There was either correct DMS, they picked an image from the same category as the stimulation, or they picked an image from a different category. That would be an error DMS. And we compared that to no stimulation whatsoever. And what we find is that the blue bars are actually fairly close to the red bars. The red bars are no stimulation, but they saw the picture a total of 3 times. They saw it once, they picked it out of a set of 4, and they told me when compared to a non-target, a distractor, or a novel image, that they remembered the one that they had now seen 3 times. With the write-in, they've only seen it twice. They never saw it as a single, and they never were given an instruction that this is what you must remember. But they picked them out as being strongly remembered, strongly recalled in the later delayed recognition, which indicates that what we did with the stimulation is as strong as the natural memory. Still can't tell you for sure if we're writing in something that wasn't there or if we're strengthening something that is there, but it sure looks like we're writing in a code, which would be really, really useful in circumstances where hippocampus is damaged, badly damaged, or absent. And as we look to fixing broken human memory, that could be very important. But it also has some important implications for brain-computer interfacing. A certain billionaire who just bought a certain social media company who would like to know when we can download our consciousness into a flash drive and load it on a computer. A number of other science fiction-ish entries for those who like cyberpunk and the William Gibson and Sterling type of fiction. Okay, we're going to integrate you with a computer, and well, being able to do this type of stimulation has some interesting implications for being able to do that. But that isn't always what we want to do. We want to help grandma remember to turn off the stove. We want to help people remember to lock the door, to take their meds, especially in circumstances where they are starting to lose enough of their memory that they can't draw a clock face from memory. What do we do to help them keep from losing it even more? And the answer, I'm getting ahead of myself a little bit. So in the waning days of the DARPA RAM program, we were also asked, what else does this work for? And the question was, can these applications, can this stimulation work not just in a patient who has epilepsy our goal would be to go into patients with Alzheimer's disease, but would it work if someone has a brain injury, if they have a stroke, if they've had repeated concussions? And so we've actually done a sort by the history of brain injury. We called a traumatic brain injury a singular event with loss of consciousness, closed head, but there were no seizures involved at the time. That would be considered a traumatic brain injury for our purposes here. For the purposes of this study, we'd also talked about a repeated mild to moderate brain injury, which would be repeated concussion. Events that don't include loss of consciousness. They're certainly closed brain injuries, and then we would have, it could be history of contact sports, or maybe a medication caused a person to fall a lot. And one of the things that we were able to pull from all of this is, yes, our MIMO models worked facilitating memory in the patients who were diagnosed with having histories of brain injury. Our controls and our TBI patients show very, very similar results, which is to say that when we stimulate with the model, their memory, their retention improved. When we stimulated them with a non-model, a randomized or a non-appropriate category stimulation, it really didn't have an effect. The other thing we looked at was what happened with the normal memory. Are we facilitating memory at all? Is it getting better than baseline? So we sorted the patients into whether or not they had a diagnosis of a normal memory or an impaired, mild to moderate impairment of memory. If the patient had a severe impairment of memory, we did not test them. If they had altered mental circumstance of any sort, we did not test them for this. And the answer is that under both circumstances of memory status and TBI status, our stimulation models worked. Here we started seeing success up to 50% improvement in memory retention. But I got ahead of myself just a little bit and mentioned, what are we doing to look at ways to treat memory loss now? And one of the big things we're doing there is to go back to the question, what is it about our models that worked? When the model worked, why did it work? We chase phenomenology a lot. A lot of times when you're working with the out on the cutting edge and you decide to continue chasing the cutting edge, you don't do all of the background mechanistic studies, the basic science research that goes along with it. And so we backed up a step and said, all right, it worked, great. Why did it work? And we started looking at the patterns that we were delivering and the ones that had the greatest success. Because one of the things that we know about Alzheimer's disease and a number of the disorders that produce a memory deficit like Alzheimer's is that we start to see a breakdown in some of the fundamental oscillations of the brain, in particular theta rhythm. Theta rhythm is very strong in hippocampus. Hippocampus appears to coordinate theta in multiple brain areas. And we also see a loss of theta power and a loss of theta synchrony, and also theta gamma synchrony across multiple brain areas as Alzheimer's disease and as memory loss progresses. So we asked the question, is there anything in our patterns, and I show you 6 example patterns from the MIMO model of multiple trials. What does the pattern look like? And we said, hey, let's go in and look at this and do a Fourier transform and find out what frequencies are present. Lo and behold, we see these 2 peaks here at about 3 and 4 hertz, which are, the 4 hertz is right smack in the middle of the theta oscillation band. So we then asked the question, well, hey, instead of going to the elaborate model, much to the chagrin of my modeling colleagues, instead of going to the elaborate model, let's look at just stimulating with fixed frequency in the theta band. And so, again, we did a pilot study. One of the things that was a little bit different is if we're going to start studying theta, one of the questions is going to be, do you just stimulate theta? Just turn on an oscillator, stimulate at 4, 5, 6 hertz, and let it run, and then stop and see what results you get. Well, we weren't quite ready to do that, but within the structure of our study, we clustered our stimulation trials into bouts of 15. So we would have 15 no-stim trials, we'd have 15 theta-stim trials, 15 no-stim trials, so on and so forth. Repeat until we get out to 165, 170 trials. Give the patient rest, do our delayed recognition task, and the answer is we see an improvement in memory with the theta stim. But we saw two different effects. Two of our patients, the first time theta stim is delivered, performance went up. The green band is the trials that have performance, and the trials that have the theta stim, and the performance stayed up when we stopped the stimulation. Stimulate again, and performance stayed up. And after a while, it started to decay. Very important, you look right out here, the fourth time we did a stimulation actually decreased the performance from what it had been, that elevated level it was at. You'll notice there are 3 traces, and I'll get back to why there are 3 color traces on here. Control, taken from 20-some patients, and I look only at the control data, the randomized, non-stimulated data. It doesn't really show a progressive change the way we saw. So it's not, one of the things we were really worried about is, do patients just get tired? And is this tailing off because they get tired? Are the patients actually getting used to doing the task? And it's going up because there's a training effect, they're getting used to doing the task. It doesn't happen, it's fairly flat. The other pattern we saw is this one. Stimulate, performance goes up. Don't stimulate, performance goes down. Stimulate, performance goes up. 2 patients, each pattern. What's the difference? There's a couple of differences we look at. One of which might be how much are we stimulating the 2 different halves of the brain. At least one patient only had 1 electrode in. So unilateral hippocampus. We tested a couple of things to see whether that might be a consideration. And another consideration we hadn't thought of, kind of hit us in the face when we were doing the analysis. These are epilepsy patients. And some of them are your age. There's a certain recreational drug that people think will help suppress their seizures. And even if it doesn't, it makes you feel good, right? Well, we had a moderate to heavy user, and we don't know for certain, but it's a potential compound. And so, but that was one of the patients that go up, down, up, down. I've got hot off the press data. We had a patient last week, implanted a week ago Monday, did our first test a week ago yesterday. And in the first test, we did the 15 on, 15 off, 15 on, 15 off pattern. That is the blue line right here. Goes up, stays up, builds over time, persists. Even when we're not stimulating, we see a persistence in the memory benefit. Great. Excellent. What are we going to do? We test these patients twice. We usually do test, retest to make sure that everything looks good. We had a unique opportunity, and I got a call on Saturday, hey, the patient's had her seizures, you need to get in here on Sunday. And I said, good, it means they won't be getting the call while I'm in Baton Rouge, which is a great thing as far as I'm concerned. Now I've got an excellent team. And frankly, one of my postdocs has tested more patients than I have. He certainly tested more patients alone than I have. And he set it up. He also sets up and runs our behavioral scripts. He said, okay, 15 no-stim trials, 15 stim, 25 no-stim. 15 stim, 25 no-stim. Let's space out that interval and find out how long this facilitation works. Light blue line, I'm not sure how well it shows up here. So I'm just going to go point to it because initial no-stim, that's the pre. Stim, no-stim. Stim, no-stim, stim, no-stim, stim. And that's where we stopped. In those first couple of stimulations, there was no persistence. We had spaced them out. By the time we were working on the third stimulation bout, there was persistence. So we probably built up critical level. This is really important to us because the same patient showed both patterns, depending on how we stimulate. They showed both memory performance patterns, depending on how we stimulate. That's really important. If we're going to move forward and develop a prosthetic, that's the kind of thing you want to see. And once again, when you don't get the results you're expecting, go back and figure out what you actually delivered so that you can possibly move quite a bit farther forward with your research. So this is where we stand. That's the hot off the press. I'm working with a company now that would like to take off with the persistent effects of the theta stimulation. We need to do a little bit more work, but we're hoping to start a clinical trial within a year, and that will be something we're moving forward with. That company is BrainGrade right here. I've given you my COI disclosure. Yes, I do work with an outside company, but that's our next step, is how do we take all of these principles. More than that, we take the one principle of a bypass for when there's too much damage, but we take the principles of the frequency stimulation for when we're still preventing the decline. And one of the important things to consider, we're talking about using brain stimulation. How is this different from something like deep brain stimulation for Parkinson's disease? In Parkinson's disease, the stimulation is being used to break abnormal synchrony. With Alzheimer's disease, we want to create synchrony, or at least boost synchrony. Very different. What's our key difference? The frequency with which we're stimulating. And so that is where we're headed from here. And I want to go ahead and thank, okay, I didn't put the whole crew here. My special thanks, Brent Roeder and Mitch Riley, are postdocs who helped me a lot. Brent's the one that has created a lot of scripts. Mitch is my electrophysiology recording expert who worked with me quite extensively. Sam Deadweiler was my mentor. Sadly, he has memory decline, and it is one of the motivating factors that keeps us going. And of course, my colleagues at USC who started us out with the modeling, Ted Berger, Bacillus marmoralis, and Dong Song, have all been essential in getting this going. And I would be remiss if I didn't mention the fact that we go into patient rooms. We could not do that if it weren't for our neurologist and neurosurgeon colleagues. And we have an absolutely wonderful set of EEG techs and nurses who work with us, even to the point of calling me up on a Saturday and saying, hey, patient's going to be explanted on Monday. You need to get your team in here on Sunday. And they're absolutely wonderful. We couldn't do this without them. And so once again, I thank you very much for the invitation. And it is great to talk with you all. I am prepared to answer questions for as long as you're going to ask them.
Thank you so much. That was wonderful. I guess we can go over here and have a seat for some Q&A.
Do that. I will bring the clicker just in case.
Yeah. Can everybody hear me all right?
Yeah.
It used to, before the pandemic, I never could have projected my voice like this, but those HVAC things in the classrooms have really helped me learn to do that. Okay, so I'm going to take advantage of having the floor to ask the first question. And it has to do with the interplay between this kind of translational, this application and the basic science. Because I guess it made me think, because I kind of often think about this as we have to really do all the basic science and then you can do the translation when we really understand everything. But it strikes me that some of these unexpected findings or these little surprises and realizations could be a great way to figure out what we don't know yet and to come up with new hypotheses for basic science. Do you find that to be?
Yeah, so every time we find something doesn't work, and we go back into digging out why didn't it work. Well, actually, one of the biggest questions we had is, why did MIMO work? I told a graduate student, probably about 5 years before we even considered doing the stimulation, the graduate student says, hey, you're recording these neurons while this behavior task goes on. What if you stimulate? It's not going to work. We're not stimulating enough places. We're not reaching enough neurons. It's never going to work. And it worked. I had a DARPA program manager say, you ever tried recording from a rat that knows the task and stimulating a task in a rat that doesn't know the task? It's not going to work. But wouldn't it be neat if it did? Okay. Wow. Yeah, and it did. And so that was one of our earlier papers, and one of the reasons why we keep looking for, is there anything generic? Is there anything common in these codes that would allow us, if you're going to build a medical device, you don't necessarily want to have to teach it from scratch with every subject. You want to have a few things already loaded that you just have to tweak. And that's actually one of the reasons why you push forward when you get a phenomenon and you say, okay, let's chase this phenomenon, and sometimes the basic mechanisms get left behind. But then you have to go back and fill in the basic mechanisms.
Other questions?
Thank you so much. This was very, very interesting. One thing that I was wondering, and it started when you were talking about the clock phase. I have worked with hemisphere and clock patients, and they have similar problems. So what I was wondering, if maybe the stimulation is not so much improving memory, but is improving the focus of attention. And have you thought about that, or why is it not? Why is it memory? Or
well, one reason why we don't think it's just attention. I think attention is a major factor. And I actually, for a moment there, I thought you were going to go into in another direction, was were we boosting the intrahemispheric communication, which is along that line as well. One of the other approaches that says, okay, what is the state of the brain overall when memory is successful versus when it's not successful, is very difficult to tease attention out of that, or focus. But one of the reasons why we don't think it's just attention has to do with the fact that there are stimulations that don't produce facilitation, that don't improve memory. When I mentioned the randomized stimulation, we use the same number of pulses with the same underlying frequencies, producing the same charge balance as the MIMO pattern, and they don't have the effect. So if it were strictly an attention boost by virtue of delivering the stimulation, then that wouldn't be the case. Now, it could still be an attention boost, and it might be a focus boost. And we can't rule that out. But that's useful in and of itself because individuals with impaired memory can pay attention, and the stimulation should not be improving their retention in that case. There's a couple of different tools we could use to tease that out. It's a really good point. I've already said in conversation with folks today, we're all coming away from this experience with great ideas. I'm taking ideas back, and I know that at some point one of my postdocs will be watching this stream, and he may even be watching it now, and he's saying, okay, Rob, you missed that one, but here, I'll tell you how we're going to fix that. So it is something, it is worth looking at. It's a great question.
I guess if I could just piggyback off of that, somewhat related, because I was curious about the MIMO and how unique is it and specific is it to the hippocampus versus is it a model that could be applied to adapted for other brain regions, or is there something special about the hippocampus?
What was special about the hippocampus for MIMO is we have two very well structured cell layers that have an input-output relationship. But that doesn't mean it's the only place it's applicable, because anywhere you can start looking at a nucleus or a concentration of cells that are going to have an ensemble input, and you can record downstream from that for the output in order to train the model, the MIMO should work. And one of our goals has long been to move upstream to entorhinal cortex and downstream to entorhinal cortex, different layers in this case, or even going as far as the prefrontal cortex and putting together some models in which you would record from one, stimulate the other. We've done some preliminary work with that in the non-human primates, but we think that the MIMO model actually is applicable to that as well.
I was interested in the differences between your categorization of the different images on, I think it was slide 19, and I was wondering, like, why would different categories be more prone to improvement than others? And the categories that showed the most improvement were animals, tools,
Right.
and plants. And I feel like those are more rudimentary things that we've shared a longer evolutionary period of time with compared to complex structures like buildings and vehicles. Do you think there could be something there?
There could be something there. One of the caveats with respect to the tool category is, again, we saw lots and lots of images that were brightly colored arts and crafts supplies. So color, one of the things we would need to tease out, we have come to the conclusion ourselves that the categories that don't work because we've miscategorized the images we're putting into them. So what we're calling a building.
Right.
And it's a messy category. Building included houses, churches, skyscrapers, also pieces of buildings, doorways, bridges, arches, a number of things like that. It's a messy category. And we really started seeing most of this effect after we had already pushed forward. And in some of the later tests and analyses, we simply dropped the category.
Gotcha.
Or we gave it, but we didn't include the category in any of our other analyses. Now, for the data mining meta-analysis, we're definitely keeping it in there, because what we want to know is if the plant category STEM facilitated a building picture memory. How many plants were in there? How prevalent were the plants, the grass, the bushes, the trees in the image? If you have a picture of a dog catching a frisbee, but on a nice green field, how much of that is plant? Or how much of it is green? I mean, that's the other question we've got. So it's something that's being dug into right now. And it's going to be somebody's nice little master's thesis when they're done, too.
Thank you.
Bree?
Yeah, so earlier in the talk you mentioned one of the goals here is to help grandma remember to turn off the stove or help people remember to take their medication. And unless I missed something, these tasks are all matching and recall. And I wonder, do you have any prospective memory tasks that you use to see, like, okay, okay, if in the future this is an effective treatment, is it going to help people with prospective memory problems?
Well, that's actually where we're going with the concepts behind the theta rhythm. And for prospective memory, the interesting thing about the category stimulation, the memory decoding model is specifically designed to, what is the pattern that says turn off the stove? Or more importantly, I turned off the stove. What's the memory, what's the pattern that says take your pills? For the Department of Defense, one of the things I've said is, how about the commander who has to keep track of everything that's going on? Can we have some sort of a nanny, some sort of a reminder that would say, hey, it's time to do this? Frankly, for professors, hey, it's time to eat. You know, you've been working in the laboratory or you've been writing this for so long, you haven't hydrated, you haven't eaten yet. So there are lots of circumstances we can imagine where the various different types of memory. There's also questions of where's the recall? What are we doing about retrieval, period? And we're not really looking at retrieval yet. We want to. It's way more than what I'm ever going to cover in my career. So the whole point of having talks like this is, if you've got an idea, run with it. Really, I truly, I mean, if it inspires you and this is what you want to do, then I'm going to say more power to you, go run with it. You may have to contend with arguing my postdocs, but that's okay. It's healthy.
Chris?
Yes, so when you're talking about stimulating the hippocampus for the pattern that's turn off the stove or for a certain conceptual category, that just raises to me this other dimension of changing in thought around the hippocampus. Previously it was, it's totally sparse encoding, episodic memories. There is no conceptual structure there, but you're remembering, discrete events. And as you're talking about it, there's an objective to learn sort of a conceptual map of the hippocampus. So I guess has engaging with the hippocampus in this way changed your thinking about what memory is in the hippocampus? And do you feel like it stands in opposition to certain sparse encoding theories of the hippocampus?
Well, as a matter of fact, that revelation, that change in thinking occurred even much earlier than this. When I started off studying hippocampus, it was place fields only. And not only that, but there's no topography. I mean, you look at sensory cortex, you look at motor cortex, and you can say, okay, if I go from here to here, I'm going from head to toe. Actually, it's the other way around, going from toe to head. And what does that mean in terms of there's no topography in the hippocampus? One of the hardest papers that Sam Deadweiler and I ever had to get published was, hey folks, we found a topography. And so our theory is that the hippocampus self-organizes. The hippocampus is a blank slate that is able to self-organize for the cognitive task at hand. And it does so through a lot of recurrent connections. And the topography we found is that in rodents that were doing a left-right delayed non-matched to sample task. And in that task, there's two levers on the wall. We extend one of them, the rat pressed the lever. We retract the lever. He has to turn around and go to a photo beam, break the photo beam, institute a delay. The rat had no idea how long the delay was, but when we turned off the cueing light, that was, okay, turn around, two levers have just come out, go press the opposite lever. And we found that there were a number of different firing patterns of the hippocampal neurons. Some of them fired in response to the sample. Some of them fired in response to the non-match. Some of them fired to anything on the left. Some of them fired to anything on the right, which, well, that's actually what we expected from place cells. We didn't necessarily expect that sample versus non-match would be discretely different, because on any given trial, left could be sample or it could be the non-match. That could change. And more than that, we started finding cells that were left sample or right sample or left non-match or right non-match, and even more surprising, left sample, right non-match, a correct trial, or right sample, left non-match, the other type of correct trial. And then when we looked at across 40-some rats, we looked at the mapping in hippocampus of where we found all of those correlates. Left, right, left, right. Sample, non-match, sample, non-match, sample, non-match. And all of those complex combinations were on the margins between them. And so we found a topography. And more than that, the topography wasn't there to start with. It formed after the animal started working the task. If we took them out of that task, put them somewhere else, the topography went away. So I think hippocampus, in terms of memory, is programmable to be whatever it needs to be for whatever task it's running. That's a long answer to your question.
No, that's fine.
But yeah, it does, it's very different.
Alex?
So on that same note, then can you, I don't know, can you tune the hippocampus to the MIMO model when you train the brain to the model so that it doesn't have to learn every single concept?
So that's actually part of what's behind the idea of the memory decoding model, because the very first review critique we got back when we wrote this up and submitted it was, how do we know that you haven't simply trained the animal to know that this stimulation is left and that stimulation is right? And of course, the answer to that review critique was we could scramble the MIMO model coefficients and have no effect on the behavior. And the other one being that the same stimulation is one trial type in one context and the other trial type in another context. So could we train a brain to respond to different patterns coming out of either the MIMO or the memory decoding model or something like that? I think we could, and frankly we have evidence for that from a prosthetic study. The studies at University of Pittsburgh with the quadriplegic who has the motor cortex implants that control the robotic arm. And I know of two subjects who have had that. I don't know if I don't know what their count is up to, but the stories that came out from that laboratory said we didn't put the electrode in the place for the wrist neuron. The patient learned to fire a pattern that would result in a wrist rotation or a wrist movement. And so the short answer to your question is, yeah, I think it's possible. And it's definitely something worth digging into a little bit more. And ultimately, as we start turning these types of medical devices, neural devices, into actual medical treatments, that's exactly where we're going to be. It does bring to mind a certain science fiction story by the name of the Terminal Man, and we don't want to go there, that's for sure.
Yes?
I first of all want to thank you for your fabulous talk. My question was actually going to be about the Terminal Man.
Oh, okay.
So I was like, this is a perfect transition.
Okay.
But I don't know if you're allowed to talk about some of the possible adverse consequences that you might see in your research because of brain grade. But I mean, are are there, I know that earlier in the talk you did say that in some of these studies there weren't serious effects, and you're very optimistic about your research, and I think it's very clear why you're optimistic, but I mean, are there any background concerns? And I don't want to bring up the exact things in the terminal
No.
there, but-
So, all right. So here's a question that came up in talking to the grad students this morning. Do we consider the ethical consequences of what we do? And the answer, of course, is of course, yes, we do. And I have something I have referred to as Science Friday. We've now shifted it to Thursdays, in which my postdocs and my grad students and I sit around and we talk. And it doesn't have to be any given topic. And we do a lot of responsible conduct of research conversations in this. There have been instances in working with the various funding agencies and meetings. Okay, what are the ethical, legal, and social implications of what we're doing? But what are some of the risks? Well, the number one risk that we would see would be inducing a seizure. Okay, so the first thing we had to do is we had to convince our IRB that the stimulation we use is safe. When a neurologist is doing cortical function mapping in order to determine whether surgery is going to affect an essential function, they use a cortical stimulator that starts at a particular milliamperage and goes up, and they stimulate an area. Okay, do you hear anything? Do you see any? Does it trigger any memories? Do you smell fudge when there is no fudge? Can you hear the color blue? And some of these synesthesias and the like. They go through this whole thing. Okay, stimulate this area, the fingers started to twitch, thumb started to twitch, and map all this out. Well, to start with, we're working. That's the minimum level that one of those stimulators could generate is 10 times higher than the maximum amperage that our stimulator will generate. So we're working with a safety margin there. The other thing we do is we do a safety test in which we ramp up our stimulation through a number of amperages and up to our target, and then stimulating one or more electrodes at a time up to our target. All of this to determine, is there anything detectable? We want to make sure that the patient doesn't actually detect anything when we stimulate. So there's a number of safety things we go through with that. We work with the patients when they're still on their anti-seizure meds, or at least still under the influence of them, because we don't want anything to happen. There have been many instances where we have walked in and walked out and said, we're not going to stimulate. And a couple of times we've taken the neurologist by surprise, because the neurologist was about to come in and tell us that it wouldn't be a good idea to do this, and we already told him we're not stimulating today. So we're very, very safety conscious. We're infection conscious. As you move forward to developing an implantable device, you have to be infection conscious. We're heat conscious, and in design considerations, you want to make sure that we're not generating more heat than what the body tissue can dissipate. Those are the physical constraints. We are aware of a number of the other considerations and always keep that in our mind. We feel ourselves constrained to the Hippocratic Oath, first do no harm, as well. But there's an even broader consideration that isn't what you've asked. Sooner or later, as we demonstrate success, individuals are going to come to us and say, hey, I want that. And they won't have the medical indication. If we come up with something that is beneficial for Alzheimer's disease or for stroke or traumatic brain injury and so on and so forth, and it restores memory function and there's a hint of enhancement, then sooner or later somebody's going to say, well, I just want to have that done anyway. I want to improve my memory. Best example is the drug aniracetam, or as most folks know it as, Limitless. The drug involved in the movie and the TV show Limitless. And it has serious side effects. And yet there are individuals who have taken it electively. And so when it comes down to a question of when do we as a society decide that a person can elect to have electrodes put in their head? And how much of that is an individual decision and how much of it is a societal decision? We have a case of a celebrity who elected for a radical double mastectomy because of family history of breast cancer. And that is not quite the same category because we're talking about trying to prevent a medical complication later down the line. But what if the person says, I want this implant because I know I have a family history of Alzheimer's and I want to head it off? I'm kind of talking around the more sensitive areas. Generally when people say, well, do you do such, I say, we don't do that. But the reason we don't do that is because it's on our mind as we move forward with this. The ethical, the legal, the societal implications of what we do is something I internalize as we move forward with this type of work and development. And it's something that I teach all of my students and anyone that works with me. And so it's not a bad question at all because it's a great way to probe for what's the personal attitudes and what are the, is there a line you won't cross? And frankly, anything that's going to cause harm is the line I won't cross.
Yeah, that makes sense. I mean, but could you envision a future where this would be something that people could just, in terms of, just thinking in terms of normal aging and age-related memory loss, just, as you get older, maybe you'll get a hearing aid,
Right.
maybe you'll get bifocals, maybe you'll get a hippocampal implant instead.
Right. I could actually see that, and I could see it being elective.
Yeah.
And but that's much more a decision for y'all than it will ever be a decision for me.
Did I remember that you had a question, Paul?
Yeah, your theta rhythm slide reminded me of a paper that came out from MIT where they used like flashing lights with an Alzheimer's mouse model, and I was wondering if you thought that what you're seeing there is related to that phenomena. And
There's a number of interesting papers that have been done in trying to train the theta rhythm using external means, using either light or sound. And there are some suggestions, especially in the rodent models, that you can entrain a rhythm and it will have a beneficial effect. But we don't know if it persists. And that's actually going to be one of the key questions. Flashing light is something we would never do with these patients for many obvious reasons. But so then the question is auditory feedback, sound feedback. We know that you can use visual and auditory and entrain an alpha rhythm. And I mean, that's been the cornerstone of neurofeedback for decades. So now then the question is going to come about is how, when, and where do we start non-invasive entrainment techniques? How focused does it have to be? Is transcranial magnetic stimulation enough? Is transcranial alternating or direct current stimulation enough? How focused does it have to be? Do we still have to get our point of focus all the way down to hippocampus, or worse yet, to the nucleus basalis or to the fornix in order to entrain the stimulation? What technologies do we have at our disposal? Is it going to be an optogenetic model? Is it going to be an ultrasound model? Is it going to be some combination of the transcranial stimulations? And we're just tipping our toe into the ocean of the possibilities that come forward, because yes, we're going to have to go transcranial at some point.
Yeah, I'm wondering what the longitudinal effects of one of something, either the theta
You know, that's a great analogy. I love analogies. I work with analogies all the time in my communicating class. Because diabetic, you have 3 ways a diabetic can go. A diabetic always needs the same medication. The diabetic gets worse and has to transition from non-insulin to insulin, or you can have a diabetic who, through other mechanisms, resolves the source. Gastric bypass, Roux-en-Y gastric bypass resolves type 2 diabetes within 48 hours. Weight loss to a person like me, frankly, and would resolve it, or it goes the other way. So longitudinally, I think from a medical scientific point of view, the ideal solution would be you boost the network, it works better, and you can turn off the stimulation. I don't know that we'll ever get to the point where you can permanently turn it off, because our target is a disease. So you wouldn't put it in, stimulate, and at some point remove it. You might turn it off for a period of time and then turn it back on. But for many purposes, a stimulation that could be periodic in that you turn it on for a period of time during a day and the benefit persists would be the ideal outcome. And yes, I foresee a condition in which you have to produce more and more of a stimulation, or more and more of an effect, in order to get back to the same level. Because again, this is a degenerative disorder. And so I think the 3 different options, it has to be supportive all the time. Think of a cast, a crutch, and a wheelchair. The cast will eventually come off, but if you break your limb again, you'll have to put it back on. The crutch is something you use when you need it. You're sitting down, you don't need a crutch. You're standing, you may be able to get away with a cane if you're walking, cane, crutch, or whatever. But when you degenerate too much, you need a wheelchair. So I think actually, as we're looking at medical devices and neural implants, we're going to be seeing that exact same model. Periodic, in that it always has to be there when you use it. Pulsatile, that you can turn it on for a while, turn it off until you need it again and turn it on. And then there's going to be instances where you just have to escalate.
Could that be the case even so is a non-neurodegenerative issue with the hippocampus like an anoxic episode or other, I think like high levels of chronic stress can cause problems with the hippocampus as well.
So an anoxic episode would be the pulsatile, the periodic stimulus, the one that has to be, you have to have the stimulation whenever you're using it. An anoxic episode that kills cells, those cells aren't coming back. And so you would need the implant to be functional anytime you need the function of the cells that are gone. You're bypassing. But something stress related could easily be you turn it on to rebalance, to reestablish the beneficial brain state. And when you've got that state, you turn it off until the next time you need it. Yeah, so both of those are non-degenerative.
Right, yeah, it's exciting.
Yep, neat stuff.
Defense might be really interested in that last one.
I know.
Yeah.
I still have a good working relationship with all of my DARPA program managers, and I also have some very unusual connections into the Department of Defense. So I've got weird friends. I've got weird friends in shady spaces.
Anyone else? That would be an interesting note to end on. Yes? No, please.
I'll try to make it fast. So you were talking about how you weren't sure if you were writing in like new memory, and what I was wondering about that was, well, we all know that memories can have a pretty big influence on your behavior
Right.
or how you respond to certain things, and that made me start thinking about other conditions, other psychiatric conditions where a person suffers from an association that causes them to have negative behavior, like a person having bad associations with food or with their self-image. And it is a pretty big leap to say that this kind of research could be used to influence people's associations between things, ethically and technologically. But I was just wondering if you thought that, I guess, inducing new or different associations, like new memories, with certain maybe bad stimuli for people could help them resolve negative associations.
Staying on the ethically beneficial side, two great examples of what you're saying are in PTSD. We know that there is an abnormally strong memory association with certain events that have very strong emotional content, very strong stress content. The funny thing is we see something similar in drug abuse relapse. There are memories that have a very, very strong association, and it's hard to break them. One of the things that I haven't had a chance to talk about, because we never really chased it as far as we would like to, is that every time we generate a model of stimulation for a particular category, there are both positive correlations and negative correlations between the category and the ensemble firing. So we hypothesize that we could come up with negative codes or antisense codes. And we, again, this is hypothesis that they may weaken associations. And so an area of research that is desperately needed is to look at weakening versus strengthening association in memory with an eye to neuropsychiatric conditions in which there is abnormal memory. And a third example pops to mind, which is intrusive thoughts in schizophrenia. And so these are applications in which we would propose that a negative, an antisense, so to speak, stimulation may turn out to have utility. There's also the question of how do you erase something that a memory that shouldn't be there. And a funny example, one of the weirdest little movies, The Eternal Sunshine of the Spotless Mind.
That's exactly.
Right, and one of my perfect examples of bad movie science. They show us a static MRI image and have this little pink dot running around like Pac-Man on the MRI. And our graduate program used to run something called Neuroscience in a Movie, and we would have we would show a movie in one of the downtown public spaces and advertise the movie. After the movie, there would be a panel of neuroscience experts who would then discuss the neuroscience in it. The very first time they ever called me out was for that movie, and my two colleagues looked at it and said, I don't know where to start. I had done a lot of science fiction convention panels by that point. I said, I do. The director, the science advisor turned to the director or the writer and says, it doesn't work that way. And the director said, wouldn't it be neat if it did? And he says, or we don't care. This is visually interesting. And they just went off in that direction. But that's one of the implications of how do you do the memory clearing, how you do the memory cleaning. Antisense or negative codes may be one way to do it. There is an implication there. Again, it's a fine line, and we have to be guided by the principles of beneficence. So, and you know, that's one of the things, if you're working with human subjects, you undergo the training for the research ethics anyway. And it's one of the things that gets hammered home. And I just had to do my 23 modules 2 weeks ago, so it comes to mind a lot. You know, every time.